Hi everyone, I am Dr. Rana Maurya, Department of Orthodontics, Banarsi Das University, Lucknow. Today I will discuss about the method of studying. This is very important aspect in the research. So there are two main approaches to studying the growth I discussed by the Prophet. These are the measuramental approach and experimental. In measurement approach, this technique is carried out on living individuals, which may be animals or humans. So this approach itself will do not harms animals and humans. So these will be available for the additional measurements at another time. So animal will not sacrificed by the experimental approach. So by this we can study the longitudinal methods, both cross-sectional methods as well as semi-longitudinal studies. So measuramental approach, it may be of three types: craniometry, anthropometry, and cephalometric radiogram. In craniometry, cranium means skull, metry means measurement. So in craniometry, it is the study of shape and form of human head or skull. Craniometry is one of the earliest approach of anthropology. So in this, measurements are done on the dry, dead human skulls, using a landmarks on the skulls, and then measurements will be done. So by this, we directly measurements the various parameters of the cranium. So it is possible to compare one skull with another and to make statistical comparison between populations. So it have advantages that precise measurement can be made by this method. But disadvantage is that longitudinal study is not possible, because in longitudinal study we study on the same persons for a prolonged time of period of time. And also this growth study can be done only cross-sectional by this craniometry. Next is anthropometry. Anthropometry refers to the measurement of human individuals. It involves a systematic measurement of physical properties of human body, size and shape, like height, weight. In this, the various landmarks studied on the craniometry or dry skulls, they are transferred to the living individuals by using soft tissue points overlying the bony landmark. Like point A, which is the deepest point between the anterior nasal spines and the alveolar kiss between the central incisors. So these hard tissue points were transferred over the soft tissue points and we studied. Yeah, we measure, take the measurements. Advantages: studies can be done longitudinal because there is no need of sacrifice of the subjects. But the disadvantages is because of the soft tissue thickness, over bone, precise measurement cannot be done. And soft tissue thickness varies over place to place. So precise measurements is not possible by anthropometry. Third is cephalometry. So cephalometry, it is standardized radiographic technique which was introduced by Broadbent and Hoffer in 1930. So this cephalometry, it utilizes the combined advantages of both craniometry and anthropometry. Means we can do the longitudinal study as well as cross-sectional study by means of cephalometry. In this, the direct bony measurements can be done on the radiographs over a prolonged period of time for the sample levels. So we can study the longitude methods. The disadvantage is it produces a two-dimensional representation of three-dimensional structure. So this is a major disadvantage. The second method after measurement approach, the second approach is experimental. It uses experiments in which growth is manipulated in some way by administration of some chemicals, some drugs, or by other methods. So this method is destructive technique, where the animal that is under observation is sacrificed. So due to destructive natures, this approach is not carried on the humans. It is restricted only on the non-human species. Experimental approach are of four types: vital staining, autoradiography, radioisotopes, and implant radiography. In vital staining, it is originated by John Hunter. In this, the growth is studied by observing the pattern of stained mineralized tissue after administration of certain dyes to the experimental animals. So these dyes which we administered in the animals, they remains in the bone and tissue where mineralization are taking place, and they can be detected later by sacrificing the animals. And after sacrificing the animals, the slides were prepared, the tissue sectioning were done, and slides will be prepared, and then we observe the area where bone growth are taking place. So possible, this vital staining is possible to study the manner and type of bone laid. So with this vital staining has to study the site where bone formation is taking place, direction, duration, and amount of the bone laid down at different sites. This method is not possible in humans because the experimental animals has to be sacrificed. The various dyes used are alizarin red, acid alizarin blue, tricone blue, tetracycline, and lead. Belgier 1936, he accidentally discovered that bones of the animals that had eaten madder plant, they were stained red. So after observing this, he discovered and the alizarin dye from the madder plant. And other dye is tetracycline, which is antibiotic, is an excellent vital stem that binds to calcium at growth site in the same way as alizarin. That next is autoradiography. In autoradiography, in this technique, a film emulsion is placed over a thin section of tissue containing radioactive isotopes, and then is exposed in the dark by the radiation. In this technique, first we administer the radioactive isotopes to the animals, and then we make a thin sections of the tissues, and then it is exposed in the dark by the radiation. So after the film is developed, the location of radiation indicates where the growth is found, because that area they get the radioactive isotopes, by which we can locate the area where growth is taking place. That can be observed by looking at the tissue section through a film. These are the methods. First, the injection or oral administration of radioactive tracers in the lab animals, and then sacrificing of animals, and we make a tissue sections of 20 to 50 micron thick for the microscopic evaluations, and the tissue are mounted into the glass slides and were embedded in a photographic emulsion to generate a latent image. And after latent image formations, the image were developed, and arrest of image developed by exposing the slides to a fixating reagent. And by analyzing histological section, we can see the area where the growth are taking place. In this, we administered the dyes to animals, and then there is a sectioning of the tissue, sectioning, then radioligand equilibrium bindings, and washing up PFA fixation and exposure, phosphorus imaging scans, then digital auto radiograms. This shows the area where bones are taking place, and then the quantification of how much bones are forming. Next is radioisotopes. We know that the radioisotopes, they have a element which have the same atomic numbers with different atomic weights. And these radioisotopes, they emit the radiation. And these radioisotopes, they are unstable form of an element that emit radiation to transform into a more stable form. The various radioisotopes used are technetium-43, calcium-45, and potassium-32. First, radioisotopes, they injected into the tissues. And after a time, these radioisotopes, they get incorporated into the bone, which were detected by tracking down the radioactivity they emit. So these radioisotopes, they act as a in vivo markers. Next is implant radiography. The implant radiography was first introduced by the Dior in 1960. He introduced this technique to study the bone. In implant radiography, It involves the implanting of small bits of biologically inert fluid, that is tantalum, into growing bones. The diameter of this yellow was 1.5 millimeter length and 0.5 millimeter diameter. So these implants serve as a radiographic reference point for the serial radiographic analysis, by which we can study the amount, direction, and manner of bone growth. These implants were placed in a certain area of mandible and maxilla. In maxilla, in hard palate, one implant is placed behind the deciduous canine, which was placed before the eruption of permanent maxillary incisors. And other implants were placed just below the anterior nasal spine. It were placed after eruption of permanent maxillary incisors. And one implant on either side of zygomatic process of maxilla. And one implant is placed between the border between the hard palate and alveolar process, which is medial to the first molar. In mandible, one implant is placed in anterior aspects of the symphysis in the midline below the root tips of the incisors. And two implants, one on right side of the uh, two implants on the right side of the mandibular body, one under the first deciduous, first premolar, and the second is below the second premolar or first molar. And one implant were also placed on the external aspects of the right side of the ramus at the level of occlusal surface of molars. Next is natural markers. Normal bone, they have a certain histological features like nutrient canals, nutrient canals, nutrient foraminas, lines of arrested bone growth, and certain prominent rabiculi. So these successive developmental features of bone, it can be used as a natural markers to study growth by means of radiographs. So these natural markers can be used to study bone remodeling, including the bone deposition and bone resorption. Other method include the comparative anatomy and genetic studies. Then methods of gathering data. There are three methods of studying growth. One is longitudinal study, cross-sectional study, and semi-longitudinal study. In longitudinal study, observations and measurements, they are made of the same person at a regular interval over a prolonged period. This represents an example of study on a long-term basis. Like here, group 1, group are studied at time 1, suppose at 5 years, at 10 years, and 15 years. So at all time intervals, the same persons are studied. So it is called a longitudinal study. Suppose if we need to study a growth of mandible for a child for 5 to 15 years, so it will take 10 years of study from 5 to 15 years for the same persons. So advantage of the longitudinal studies is that by regular and serial comparison, the specific developmental pattern can be studied. Means because we are studying the same person at prolonged time, so we can study the individual variations in development. So specific variation of development of individual in a group can be studied properly. But disadvantage It is time consumption is more to follow up years together, because same individuals are study for prolonged time, so it takes more time. And repeated studies are not possible in case of any. If any errors occur, any flaw occurs, we cannot go back for the study. Like when we are studying a child growth from 5 to 15 years. So at the time of 10 years, if we saw any errors or flaw, so we cannot go at 5 years, because individual it grow from 5 to 10 years. So we cannot correct any flaw. Any specific lab research tools and data storage are expensive. For prolonged study, we need to storage of samples. So it takes more lab area and is expensive. And attrition in sample size means there is reduction in sample size. Suppose we are studying a student of class 3, is comprised of suppose 60 students, and we need to study from 3 to suppose 5 to 15 years. So after 5 years, some students leave the city, some students go to other college. So there is loss of sample size in longitudinal study. In cross-sectional study, in this the group of various ages at various stage in development are examined at a time. Like here, group 1 are of different growth stage, group 2 of different stage in development, group 3 are different stage. Suppose we need to study growth of the mandible like 5 to 15 years. So we can take a sample of 5 years of child, 10 years and 15 years. So we take the samples at the same time and we study at the same time. So cross-section study, it takes less time. So it is quicker as it studies a short duration, because we are taking all the age group at the same time. So it takes less time as compared to the longitudinal study. And it is possible to get a large sample as the duration of the study is short. And also the repeated studies is possible easily in the case of NF, which is not possible in the longitudinal study. And they are less expensive because it does not require lab area for the preservation of samples for prolonged period of time. So it is less expensive. But there are certain disadvantages of cross-sectional study. Because in this study, we are comparing measuring groups of study are always not similar, so that the appreciable comparison is difficult to obtain. And also we cannot get the specific variation of development of individuals, which is possible in the longitudinal study. Overlapping a semi-longitudinal study. This method is combination of both longitudinal and cross-sectional methods. So it derives the advantage of both methods in gathering data. Suppose if we need study of growth of animals from 5 to 20 years of age. So we take two data, one group of 5 and other is 10 years. After 5 years, initially we have a two type of data, one from 5 years and one from the 15 years, sorry, one from 15 years. So after 5 years, the 5 become 10 and 15 becomes 20. After 5 years, we get 4 data. One 5 years, 10 years, 15 years and 20 years. Because initially we have two, one 5 years, other is 15 years. So 5 years, child becomes 10 years and 50 becomes 20 years. So it have advantage of both longitudinal study, because we are studying for 5 years, and cross-sectional because we are taking two age groups of individuals. And types of growth data. There are 4 types. One is opinion, observation, rating, ranking, and quantitative. In opinion, it is the oldest method and crudest means of studying growth. It is considered a person's own clever guess without any calculated data. We only guess that this child becomes that this child is growing faster as compared to the other child. It depends only on the clever guess of some persons by experience. There is no any scientific data, the calculation of data. So this is expression of person's opinion and simple observation. It is not very scientific and should be avoided when better methods are available. Next is observations. It is useful for studying all or non-phenomena. Means we are observing like absence of caries. We observe the patient and we find these caries are present or caries are absent. Periodontal disease are present or periodontal disease are absent. The patients have class 2 molar relationships, a patient have class 1 molar relationship, a patient have class 3 molar relationships. So these are based on the observation, present or absent. Rating and ranking. The certain data are difficult to quantify, and thus we may use of rating and comparison with accepted conventional scales or classification. Like in particular community for the particular age, that boy is taller. Smile comparison. We cannot compare and measure the smiles. So we make a rating scales. Like from suppose we have a 0 to 10. Suppose 0 to 2 have bad smile. 2 to 4 not good. 4 to 6 good. 6 to 8 very good. 8 to 10 excellent. So we have a rating scales on which we rate that data. Ranking. Ranking is the method of arranging the data in sequential order according to its value. The rank of students are determined and their performance in the school. Like 10 students are participating in sports. So we can arrange the performance of that student from 1 to 10. Like this child have first rank, they have second rank, they have third, they have fourth. Like 60 students are appearing in examinations. We make a report card according to the students which get the highest score. And that student that get a lowest score. So we arrange these marks according to their performance and then we rank that data. Next is quantitative measurements. Quantitative measurements is scientific approach to study growth and is one of that is based on the accurate measurement. Quantitative measurements, by this the data can be obtained direct method, by indirect method, by derived method. Indirect data we obtain from the measurements that are taken directly on the living persons or cadavers by means of scales, caliper, or measuring taps. We can measure directly the height of that person is so much, weight of that person is so much. So we can measure directly over the persons or cadavers. Whereas indirect data is measurement taken from the image like cephalograms, OPG X-rays, CBCT scans, photographs, radiographs, or dental models. So these are the analog. So in indirect measurement, measurements are taking on the analog. Whereas the derived data, the data that are derived after comparing two measurements. These two sets of measurements can be different time frames of two samples. Like when we want to know the how much child is growth from 10 to 15 years, this individual. So we take a height at 10 years of age and second at the 15 years of age. Suppose at 10 years of age, this boy had suppose 100 centimeter and 15 years of age, suppose 125 centimeters height. So we subtract 125 to 100. So we get a 25 centimeter. So this 25 centimeter is growth from 10 to 15. So this 25 centimeter, it is derived from the two sets of measurements at different times. So this is derived. Thank you. I hope that you have understand the method of studying growth in orthodontics. Thank you.